replication proceeds in both directions. And we'll talk about that in a second. All right, so first is the recognize the origin of replication, then the two strands split, and the replication forks are formed, right? Okay. The next thing is this enzyme helicase, and in the video it was like that big, like kind of like brown, like complex thing, right? But for now, just for our simplicity, I've like made it look like this. So helicase, once you've recognized the origin of replication, helicase comes along and it unwinds the helix, because obviously you know it was like already twisted to begin with, and then it further separates the two strands from each other, right? And again, I apologize for this horrible animation, but this was the best I could do with my core skills. Okay? So we're good so far? Yeah? All right. At any point, if it gets too confusing or you have a question, please come off mute and raise your hand, or and just say what, ask your question. Okay? All right. So helicase unwinds and separates the two strands. So now we have the two strands. Do you notice how the strands of the original parent one were labeled? Do you guys see that? So I want you to just keep in mind which were the original strands. You should always be taking note of where those original strands, like 5' and 3' are. All right? So then helicase. The next thing, and I don't know if this is in the video, but there are these things called single-stranded DNA binding proteins. So they're like abbreviated SSBPs. And what they do is they hold the strands apart once helicase has actually like separated them. They kind of attach there so that the DNA doesn't re-anneal, or it doesn't reconnect to itself, right? Because we want to keep them open so that we can actually add more DNA in there to make the new daughter strands. Okay, so the SSBPs, basically their role is just to hold the DNA once it's already been separated. Pretty easy, right? Okay, next step. Then there's this other enzyme, and these are all enzymes. This is called DNA gyrase. And basically, like, can someone tell me, so if I have a slinky, you know how slinkies are all like curled up? What happens if I untwist the slinky at one part to make it kind of like flat? What's going to happen at the other ends of the slinky? Anyone? Do I hear a brave person?
Wait, can you repeat that?
So if I have a slinky, and I untwist it at one part and then try to make it flat, what's going to happen to the other, like the extreme ends of the slinky?
It'll stretch out.
Do you think the other ends are going to stretch out, or do you think they're going to super coil?
If the middle's being pushed in.
No, if the middle's being unwound.
Oh, then the other parts are going to compress.
The other parts are going to be like super coiled, right? Okay, so that's basically what happens to DNA too. If you unwind one part and like stretch it out, the other ends, like on the other side, are going to become super like pressurized and tense and super coiled, right? We don't want that to happen. So there's this enzyme called the DNA gyrase, or it's also called topoisomerase. And basically, that's what it does. It relieves this super coiling, or the tension that results from the unwinding of it to begin with, right? So it's like it goes beyond the main like part of where the replication is happening, and it just kind of like makes it looser, so it's not like super coiling, because then it could be damaged or could break apart. So that's relieving the tension. Okay. Yes.
What did the blue dots represent?
Those are the SSBPs.
Okay, thank you.
Single strand binding proteins. Okay, we're good so far? Yeah? All right. So we have 4 things so far. The next thing is to remember that replication goes in both directions. We already said that, right? But just for like the sake of our like sanity, I guess, we're only going to focus on one direction, right? So I want to concentrate just on one side, right? So we're going to go like that. Are you guys all okay with that? And you see what I did, right? I'm just ignoring the other side for now. All right? Okay, let's see what are we gonna do here. Okay, so I want you guys to when I did that, when I went from this to this, right? I lost one of my ends, right? Do you see like how I lost one of my ends on each of the strands? So I want you guys to go back and relabel them. So what's this one gonna be? What end is this again? On this strand? If this is the 5', then this is gonna be the what end?
3'.
3', right? And if this is the 3' here, then this is gonna be the 5', right? So that's what I always want to do. I always want you guys to label them so you know what's happening. So I'm labeling the original ones, right? Now I want you to label the ends, the 5' and the 3' of the new strands that are going to be built. So you know that this original parent molecule is separated. You saw how it's separated, right? And now from the environment, we're going to add in new nucleotides complementary to each of the strands. So these original strands never actually come back together. That would be like conservative model, right? Which was incorrect. But they actually go into making the new strand here, and then there's going to be a new strand here. But half of the original parent was already in there, right? So can you guys go ahead and label the new strands? So if I'm going to build a new strand that's complementary to this top one right here, what end is this one going to be of the new strand?
5'.
That's going to be the 5' end, and then if I build on here, then this end of the new strand is going to be the what? 3', right? And what about down here, the one that's going to be added onto this one? This end is going to be the
5'.
5' of that new one, and then this end over here is going to be the
3'.
right? Okay, we're good, right? Everyone understands that?
Yes.
Easy, right? Okay. You guys remember what happens next? Anyone?
What is it? The primate? Like the RNA fragment?
Yes, thank you. So, all right, first I want you guys to remember that DNA can only be synthesized in the 5' to 3' direction. And I'm talking about the 5' to 3' direction of the daughter strand, right? So I'm not looking at this. I'm not going to synthesize from here to here. I'm going on the daughter strand. Here's the daughter strand, right? So it can only go from 5 to 3'. You get me? You're with me? Okay. So first I'm gonna primase comes in, right? Oh, whoops. Yes. Sorry, hang on one sec, guys.
Through your PowerPoint
Yes.
and then dialoguing with them.
Yes.
And do you ask them questions or they just ask you questions?
I'm asking them questions, but then most of them are on mute, so they're not answering. But then I wait for a few seconds, and then someone will answer. So
Okay.
yeah, so it's not perfect, but okay. Are you gonna be here after? So I'll talk to you. Okay. All right. Sorry about that. Okay. So anyway, so which one? So if I'm going from 5 to 3', and the whole replication bubble is moving to the left, right? Which strand can be synthesized continuously? If I want to go from 5 to 3, and I want the whole thing to be moving toward the left, which is going to be my leading strand then?
The one on the
The
top.
lower one.
The one on the top, right? Because I'm going from 5' to 3', the replication bubble is moving this way. So this is going to be my leading strand, and then this one down here, if I want to go 5 to 3' on the daughter strand of this bottom one, which way would it have to go? It would be going this way, right? Which is going against where the main replication bubble is moving. So this one can't be just moving, it can't be synthesized in this way, because then it will just go the wrong way of the bubble, right? Okay, so this is the leading strand here, right? And then the other one would be the lagging strand, right? Exactly, good. So there's the leading strand. This bottom one will be the lagging strand. Okay? Are you guys with me? Sorry, here, right? All right, so then the next step is that basically, so primase, do you guys remember that? Primase comes in and it puts down a primer. We're just focusing on the leading strand for now. We'll talk about the lagging strand after, but the leading strand, first of all, primase comes in, right? Here's primase, and it's going to place a small RNA primer toward the 5' end of the growing daughter strand, right? So again, these T's that are upside down, they're supposed to represent like nucleotides, right? And bases that are pretty much like they're complementary to each of the original parent ones over here, right? So here's my primer. Primase puts down a primer, right? And so which end is the 5' end of the primer? This one or this one? First one or second one?
First.
The fish one.
The first one. So the first one's the 5' end of the primer, and then that means that this is the 3' end of the primer, and then we always want to build onto a 3' end, or basically going from 5' to 3' of the daughter strand. So so first primase comes in and makes that primer, and then DNA polymerase 3, I want to call it DNA polymerase 3, adds nucleotides from 5 to 3. So watch how that's going to look. Here's DNA polymerase 3, and it's going to go like this. Are you guys with me? Yeah? And then do I have to stop anywhere on this strand? No, because I'm just going continuously from 5' to 3', like that's the way DNA polymerase can add it. So there's no problem whatsoever, and I just make this whole long continuous strand all the way to the end of the strand. Right? You guys see how that works? So the leading strand has no problems whatsoever, right? It just makes that daughter strand, and then now we have a new DNA molecule up here, right? We're good? Okay. So meanwhile, this is where it gets crazy. So just try to follow along with me, right? Remember, the whole replication bubble is moving which way? To the left, right? So we can't just say, oh, let's place a primer here and then have it continuously added on this way. That's not going to work, because then it's going to be going in opposite directions. We need the whole bubble to be moving together. So what happens is primase places a primer on the lagging strand, but it doesn't place it all the way over here. It places it somewhere like around there. Okay? Can you guys tell me which is the 5' end and which is the 3' end of this primer? Which end is this one right here? That's the 3' end of the primer, right? And this is the, and this one right here is the 5' end, okay? And which direction can DNA polymerase add nucleotides onto?
5 to 3.
From 5 to 3, or it can add it onto the 3' end, right? So here comes DNA polymerase 3. So which way am I going to add the nucleotides? This way or this way? First way or the second way? First way, right? So watch how it's happening. And I have to do it from the primer. I can't start adding them here like this. I have to do it from the primer itself. So I'm doing this, and then here's my new DNA here. Right? Do you see that? We're good, right? Now I need to add another primer. So I repeat the process. So I add another primer here, right? We're good there? And then DNA polymerase 3 comes in again, and then it adds DNA nucleotides in the 5 to 3' direction, going that way, right? Then what happens now?
Get another one.
I get another what? Another primer. So primase comes in again, adds in another primer. DNA polymerase 3 comes in again and adds what? More DNA nucleotides in the 5 to 3' direction. So the actual building of the nucleotides, like onto the little thing, onto the primer, occurs going against, it seems to be going against the direction of the main like movement of the bubble, but each subsequent fragment is going closer to the bubble. So this is like going zoop, zoop, zoop. So the general direction is still moving to the left, right? But the actual construction of each fragment is going to the right. Do you guys see that? And then this thing right here, so starting with the primer, at which end is this? Which end of the fragment is this? Right here, where my arrow is. Is this the 3' or the 5' end of this little segment? This one right here.
Prime.
That's the 5' end of it. And what's this end over here? That's the 3' end of this little fragment, right? So this one that starts with the primer and then goes toward the 3' end with DNA nucleotides, that thing right here is called an Okazaki fragment, right? And you guys have to know the order in which Okazaki fragments are made. So the first one's made, this is the first one that's made, right? And then we kind of jump over here, and then we make the next Okazaki fragment, primer first, and then nucleotides going in this order, right? And then we jump again, primase comes in, and then we add the nucleotides from the 5' to the 3' direction, right? Going this way, right? But you have to know that this Okazaki fragment isn't formed first. This is the first one that gets formed, then this one, and then this one, right? And you have to know that the nucleotides can't just be built out of nowhere. They have to be built on to the primer. And it gets built on to the 3' end of the primer, going from the 5' to the 3' direction. Are you guys following me? Yeah?
Yeah.
Do you have any questions so
Yes.
far?
No.
Wait,
No?
so what is the those like supporting things? I forget what they're called.
Okazaki fragment?
You know the SSVPs. What do they do again?
Oh, they're just holding the strands apart while this is happening, so they don't like reanneal. The DNA doesn't come back together.
All right, thank you.
Are we good so far? So you guys would be able to identify the Okazaki fragment?
Are the Okazaki fragments only on the lagging strand?
And you could think about this whole thing right here as like one sort of mother of an Okazaki fragment, right? But really, they don't call them Okazaki fragments because it's just like this one singular, nice, big piece. Whereas these, those are just on the lagging strand. Yeah, good question. Okay, so are we good so far? Are we done? Yes? No? No, we're not done.
No, we're not.
Is it enough to be twisted?
So it has to first. So now that we're basically, it just does this until it it reaches the end, or until it reaches the next origin of replication. So that so in any case, so this is sort of the general pattern, right? But the problem is, do we want RNA in our final DNA?
No.
No, it has to be replaced with DNA.
Exactly. So what is in charge of that? It's this thing called exonuclease, or it's also called DNA polymerase 1. You have to know the distinction between DNA polymerase 3, which is the one that adds the nucleotides from the 5' to 3' direction, and then DNA polymerase 1, which they call in the video exonuclease, which basically replaces the RNA primers with DNA. So check out the great animation here. What's going to happen to these little light green teas? They're going to become regular green teas, right? So now it's nice DNA. But what's the problem here? There's still one problem. These, where the primers were, and where the DNA fragments, where the DNA nucleotides were, there's still a gap there. So there's this other enzyme called DNA ligase, which basically comes in and seals. Do you see all those little orange seals that I put in there? It basically reestablishes the phosphodiester bonds in the backbone, so that there's now one continuous molecule instead of a little, all these little choppy parts, right? So DNA ligase comes and seals it up all nice, right? And then now we have two beautiful strands of DNA, right? Daughter DNA. And you notice that one was made of the original parent, one strand of the original parent, and one is made of the new DNA here. So if this were Meselson and Stahl's experiment, right? The purple would symbolize the the DNA cell from the cells that were grown on which nitrogen? Which kind of nitrogen?
15.
Nitrogen 15, which is heavy nitrogen, and then the green would be what kind of nitrogen? The cells grown on which kind of nitrogen?
Light
nitrogen 14, exactly. And then, so what would the average density though of this whole molecule that's made of half 15 and half 14 be? If I centrifuge this molecule here, where would it land up?
14.5.
At 14.5. So it would form, and you guys recognize that this whole molecule and this whole molecule are the same size, right? Because they're each composed of half heavy, half light. So even though there are two separate molecules, they're still only going to form one band on the centrifuge tube because they're still the same mass, and that's how centrifuges separate things by mass, right? Not by individual molecule. Okay? Are you guys okay with this?
Yes.
Okay. So, what I want you guys to do now, hang on a second. I'm gonna do this on the board so that you guys also have, another understanding of how to do this, right? So, just give me a second to draw it out and then sort of see if I can get,
PowerPoint.
huh?
Will you post the PowerPoint?
Yes, I will post the PowerPoint too. So hang on. Alright, so so I'm gonna go from here. I'm gonna end the show right now because I actually want you guys to watch me. Okay? And then we'll go back to that if we need to, alright? So, and I need to set up the camera so it actually looks, so you can see what I'm doing. Alright, oh, that's perfect, isn't it? Is it backwards for you guys?
No, that's good. I see perfectly.
So this says this isn't a backwards 5, it's an actual 5?
Yeah, it's an actual 5.
Actual 5.
Oh, it's weird. Mine has like there. Hold on, look at it. Myself. Anyhow, okay. So I'm gonna use some different markers. Okay, so you guys notice how I did when I just did this, I did the left side, right? On the PowerPoint. I want you guys to copy this as we're doing because I think it's a lot. And it's going to take a while for me to do this, so I apologize, but okay. So you guys recognize what I just made, right? What is this whole thing here?
It's split apart DNA helix.
This is okay. So it's a separated DNA helix, right? So helicase came in and unwound it, and it separated the two strands, right? SSBPs, let's make those like right here, let's say. They're holding the strands together so that they're no longer, so they won't just like automatically reseal because they're complementary bases, right? They do kind of want to reseal, right? Question, what was the bond that was holding this together in the first place? What kind of bonds is helicase breaking?
Hydrogen.
Hydrogen bonds, excellent. Very, very good. All right, so let's say that we're going to find the origin of replication. Let's say the origin of replication is here. Okay? And it would have been here too. So then now there's the replication bubble, right? And now I went to my left side before, right? So now I'm going to go to the right side, and I want you guys to help me do this, okay? So first of all, what was the first step that I have to do? And you guys can unmute yourselves if there's not too much background noise and sort of help out. So what do I do first? And this is the thing that I said to always keep in mind the whole time.
Label your 5 and 3' ends.
Thank you. So which prime end of this is this of the parent strand? This one is the
5 prime.
So this is the 3' end of the parent over here, right? Right? So then that means this is the 5' end of the parent, right? And that means that this is the what prime end of the parent?
3'.
3' end over here, and over there, right? Now I want you guys to label the prime ends of the new one that I'm gonna make. So what prime end of this one is this gonna of the new one is this gonna be?
3
It's gonna be 3 prime of the new one, right? And that means down here, which end is this one gonna be?
5 prime.
5 prime.
5 prime, and if this is 5 prime of the old one, that means this is gonna be
3 prime.
3 prime of the new one, and then this one's gonna be the
5 prime.
We're only looking this way for now, right? Okay, so tell me which strand, the top one or the bottom one, for the new one is just going to be continuous from 5 to 3 in the same direction that the whole bubble is going, which is going that way anyway.
The bottom.
Bottom?
Bottom.
The bottom one, right? Why? Because look, the new one, the daughter strand that I'm building, I want to go from 5 to 3 prime of the daughter strand. Okay? This is the one that will just be able to go 5 to 3 in the same direction as the bubble. The bubble's going outward, right? What about this one? If I want to go 5 to 3 prime of the daughter strand that I'm making, it would be going the wrong way, right? So this one is going to be the leading, this one down here, this one's going to be the leading strand. Again, you have to make sure we realize that it's separated. Like I'm gonna draw this line here just so we don't get confused, because the other way it's gonna be the total opposite. So this one is the leading strand here, right? Because this is the 5 prime end, and then this one's gonna be the lagging strand. Right? You guys are with me, right? Okay. So now I want to do. What do I do next? So I separated it with helicase, right? Separated, let's say like this, here's helicase. It's separating and I'm unwinding it, right? And then I have my SSBPs right here. Let's put some SSBPs over here. They're holding the strands apart and keeping them from reconnecting, right? Remember, when I unwind something, at the other end it's super coiled, right? So over here,
Gyrates.
I'm gonna have gyrates just to kind of like, just to kind of keep it from over coiling, right? And now what do I want to do? Let's look at the leading strand first and see what happens. So what do I do for the leading strand? What do I
Yeah,
put?
it's like some of the RNA primer, and then the rest is the polymerase 3.
How many primers do I need for my leading strand?
1.
1.
I just need 1, right? Which is very close to where the origin of replication started. And I go from 5 prime, and then I build on, so let's say blue is my DNA polymerase 3, and then I just build on my nucleotides, right? I'm adding like a nucleotide here each time. You see what I'm doing? Stop at any point, just until I get to the end, right? So now I have my nice continuous leading strand, right? We're fine. Good? All right, what about the lagging strand? What happened to the lagging strand? Which way? Good. I need to do it in Okazaki fragments, right? So I need to put my primer. I don't put my primer for the lagging strand right on the origin of replication. I move it to the right a little bit. So here's primase adding. It doesn't matter how many primers, how many bases are in the primer either. That's like very random. You can do it whatever way, whatever number you want. So here's my primer, right? Let me label my ends of my primer. Which is my 5 prime end of the primer and which is my 3 prime end of the primer? Which one is this?
5.
This is my 5 prime end of the primer, and then this is my 3 prime end of the primer, right? Which end am I going to build on to make the Okazaki fragment?
3 prime.
Build onto the 3 prime end, and then I have to build toward, right? So, in what general direction does each subsequent nucleotide get added on the lagging strand? Does it go toward the replication like toward the general direction of replication or away from it? Are you guys writing stuff?
It's kind of lagging. So like,
Oh,
yeah, the thing's lagging.
lagging?
Yeah, a lot.
So can you repeat the question?
So the question was, so each individual nucleotide that I add on, is it going toward the replication fork or away from the replication fork when I'm doing the lagging strand?
Away.
It's going away, right? But in the leading strand, each subsequent nucleotide is being added toward the replication fork, right? So you see how I made my primer here, and then I built 1, 2, 3, 4, 5, right? You guys all saw that, right? So it's kind of weird because it's getting synthesized in this direction, whereas this whole thing is getting synthesized in that direction, right? And then what about the next one? So what happens after that? I have to take another primer, right? And it kind of jumps down here like, let's say like there, right? So there's my new primer. And then which direction are the bases going to be added? To the left or to the right?
Left.
To the left, right? And I don't want, so, and also in your videos, when you guys make your videos, I don't want you to just put the primer and then have all of the DNA just suddenly appear there. I want you to show the actual like sequence that one is getting put on here, right? And then another here, and then another here, then another here, and then another there, right? Because if I just see like all the primers show up and then all the Okazaki fragments show up, then I won't see that you know that it's actually going step by step back this way, right? So I need to see each nucleotide be added in when you do your videos, okay? So keep that in mind. All right, then what do I do? Again, are you guys sick of this yet? So I take another primer over here, right? And then which way do I build it? The left, right? So here's another DNA, right? Right? Do you see what I'm doing here? So again, each time I'm building the DNA going this way, but each subsequent Okazaki fragment is going this general direction, right? So it's like hopping to try to, and this takes a little longer to do than this one does. That's why that one's called the lead. That's why this one's called the leading one, and that one's called the lagging one because it's going slower, right? Are you guys all with me? Okay. Okay. So let me just. Okay. So let me also. I want you guys to look at this side again and see if you can fill in that side. And like this is pretty much based on what the PowerPoint was. But I want you guys to notice one thing. So in this case, I want you to label the ends again, right? So this is the 5' end of our original one, right? Of our original parent strand. What end is this of that strand? If we're looking this way. And this is the 3' end of this one, right? And what about here? This is the 3' end of this parent strand. What end is this here? This is the 5' most end of that one, right? So if we're going to do the daughter strands, what's this one going to be of this daughter strand? 5', and then this one's going to be the 3', right? So, and then this one's going to be the 5', right? Because 3, 5, and then this one's going to be the 3', right? And then which one is going to be the leading strand in this case then? The top one, right? And this is something that I want you guys to sort of recognize, that on either side of the replication fork, the leading strand and lagging strand are like kind of like catty corner to each other. It's not like symmetrical like this. If this is the leading strand on to one side of the replication origin of replication, this is the lagging strand of that same one, right? So in this case, again, we're going to add on nucleotides, right? one by one. You guys see where I'm doing this so like inefficiently, just to show you that each one is getting added on like in that fashion, right? And then for the lagging strand, again, this is what I want to see here. So here's the primer. And then I'm doing 1, 2, right? See how I'm adding those on like that? Another primer. Adding, right? Adding. Another primer. Adding. Do you guys see what I'm doing? So the general like direction of DNA synthesis in this one is going which way? This way or this way? Left. To the right or to the left? It's going to the left, right? But the general like direction of each fragment is getting built in which direction? To the right or to the left? Huh?
Right.
To the right, right? Okay, I have a question. So on a test, I'm asked to circle an Okazaki fragment, right? And I do this. Is that right or wrong?
That's wrong.
Wrong.
Okay. Why is that correct?
Because that's not like the right primer.
Exactly.
For that one.
This primer did not build on to here, right? Which DNA came from this primer?
Like the one at the very end.
The one at the very end, right? So I need to make sure that we don't make this mistake, and I make it clear that the Okazaki fragment is that thing right there, right? So you guys are with me, right? Here's another Okazaki fragment here, and then here's another Okazaki fragment here. And the same thing with this one, right? So if I wanted to show my first Okazaki fragment here, it's not this, right? Make sure you know which primer and which actual DNA was built off of those respective primers, right? And make sure you can identify which Okazaki fragment goes with which primer, okay? Are you guys good with this or no? So on your video, so I'm gonna give you tomorrow. For tonight, I want you guys to just try to remake something like this, just on your own, like maybe on a piece of paper, so that you guys are prepared to actually like when you get into your groups tomorrow, make sure you're prepared to actually like, start thinking of ideas of how you can represent this in a I don't want it just to be on a whiteboard and drawn out. I want you to do some sort of like video where you can kind of visually like show how this process is happening, and I want you to show like individual things being added on. Again, sometimes in the past, groups have just like kind of had like one frame where they have like no primers, and then they put all the primers like just in one shot, and then they put all the DNA, but they don't show how the DNA was actually being added on to each respective primer. So make sure you think about ways that you could do that. So I don't know if you can do like something like stop motion, or if you can do something like, even if it's like a slideshow or like some sort of animation, like, on some app. You guys know more about that stuff than I do. And even if it's just like taking little pieces of paper and like kind of dragging them along, together, I think that, I want you to get the feeling for like how this process is actually going. Okay? Are you guys all right? Are you with me? Okay, so for those of you, when I comment on your homework, I would like you to resubmit it. Especially for people who like were missing some whole page or something, I can correct. I'll adjust the score. Most of you guys that I saw so far were pretty good. Like your homework is pretty good. Then there was like if there was like some questions that were actually like answered wrong, I gave you like a 19 out of 20, which, so it was that you did it and you put a lot of effort into it, and it was, it was good, but then maybe there was like a few inaccuracies. If there was like a whole incomplete part, I gave you 15 out of 20, but if you then resubmitted that part, then I will adjust that grade and stuff like that. and then for the rest of you, I haven't gotten a chance to look at yours yet, so please, I'll return those to you today, and then please resubmit it, in the correct, like with the corrections. And also, when you guys are taking the pictures with your cameras of your work, I was able to get most of them, but can you like try to make it a little more exact, like with the frame, so there's not too much like weird background stuff, because it is hard for me to read some of them. So try to like zoom in and like make the the borders, I guess, as neat as possible, so I can actually like see it a little clearer. So that's my other request. So the worksheet for today, I'd like you to finish it and then send it to me again, because we just need like records that you guys are doing something every single day, right? And tomorrow I'll introduce the this project, the video project, and I'll break you guys up into groups. And I do want to see your group, so we'll make the different hangouts for all of our groups and like try to figure out how we're going to approach this project. It could be a huge disaster. We might need like 3 days instead of 2 days, but we just need to communicate about it and make sure we're all at least on the same page, and that we know like what needs to be done and stuff. But we do, and in terms of like assessments and stuff, this was usually like a secondary assessment, but the point of assessments is just like whether you guys understand the material or not. So if I can get a clear enough picture, like from my observations of your group work, and like random questions that I ask you while you're doing your group work, and your final product, then maybe this could take the place of a primary assessment, or I can use that as a primary assessment. I'm not really sure how the whole assessment thing is going to work. I know that the principals are supposed to meet this, I think today, and figure out a way to do that, but it's still uncertain. So, I hope that you guys don't end up getting slammed at the end of, whenever we come back to school, like having like all these, tests and stuff. But, the reality is that might very well happen. So, the best thing is just to be prepared as possible. So just, be ready for anything, but just make sure that you're reviewing in advance and stuff like that. Huh?
When are we coming back?
That's a good question. I have no idea. Who asked that? Who was it? Arya? Arya Patel. Okay. No, I have no idea. Today's the last day that we're actually allowed to be in the building. So, again, yeah, in the next few days you might see daughters running around my house, or dogs, or my husband, just be ready for anything. So, yeah, I'm not really sure. I think that the goal was 2 weeks. I read that, New York, is it the 20th? 8 weeks? Some people are saying 8 weeks. I heard that Canada closed until like September. So, yeah, it's a developing situation. So I know more than you guys do. But, I'm ready to teach you guys this way, and I think it's pretty good that we have the technology, right? I mean, do you find it a big difference from actually being in the classroom, or?
I actually prefer it.
You prefer this?
Yeah.
Because you can wear your, like, native headgear?
Yeah.
It just feels weird.
I think it's pretty cool.
Because it's like you're learning at home, and I'm so used to having a different place to like learn and relax.
Yeah, I don't know, personally, I'm not very good at like studying when I'm at home. So I think that, it's going to be a different, you have to change your mindset. I mean, if I hear my daughter screaming in the other room, I don't know if I'm going to be able to like really concentrate that much on, trying to teach you guys. I mean, I hope, and I'll try, and I'll tell my husband to like keep them, like outside or something during our class. But, the fact is, like, I have to actually teach them too. Because they're in kindergarten. Well, one's in kindergarten, one's in pre-K, and the teacher like posted all these things that they have to do, but like they get a vacation, But like I have to like teach my daughters, which is like worse than teaching you guys. So not that that's a bad thing. You guys are great. But there's a lot of like unknowns here that are going on. But But on the plus side, you guys are learning through history, so someday you can tell your kids and your grandkids, like, oh, COVID-19, like, I was around for that, and, like, that revolutionized, how people, live their lives and speak, and, I don't know, it's going to be interesting. So, anyone have any questions or anything about their homework?
Oh yeah, I didn't get number 11, which is what are three differences between DNA replication and prokaryotes?
I didn't do that yet, actually. That's a good question. I'm gonna post it on I'm gonna post the PowerPoint on Classroom, but I'll answer that question right now. One difference is that prokaryotes have one origin of replication, and eukaryotes have multiple. Right? And the other difference is that prokaryotes, it occurs in the cytoplasm because they don't have a nucleus. And in eukaryotes, where would it occur? In the nucleus, because that's where the DNA stays. And then I think the other difference might have been that eukaryotes have a single prokaryotes have a single circular chromosome, whereas eukaryotes have multiple linear chromosomes.
Okay, thank you.
Alright? Alright, so yeah, when you guys are done, submit that, and then I'll check it off. And if you have questions, like if you have no idea what I did here, if you don't understand the banding pattern that I have to install, we can do a separate Google Hangout at some point, but you have to let me know so we can, arrange it. And there might be other people in the same boat, so don't be shy about reaching out. Okay? All right, good. Well, have a great day, everyone.
Thank you.
Is the drawing homework? Like, do we submit that somewhere?
Is the what? No, the drawing is just so you can kind of like keep it all in order, and so that you can prepare to be a better contributor to your group. So you don't have to do that. If you want to send it to me and have me check it and make sure that it's right, feel free.
Okay, thanks.
All right. Have a great afternoon, guys.
Bye.
All right. Bye. Bye.
Bye. Bye. Good night. Wow.